அனைவருக்கும் வணக்கம். பயாலஜி தமிழ்நாடு சேனல் சார்பாக உங்களை அன்புடன் வரவேற்கிறோம். இன்றைக்கி நம்ம என்ன பார்க்க போகிறோம் அப்படின்னா, உங்களுடைய ஸ்டேட் போர்டு Botany இங்கிலீஷ் மீடியம் புக்கில் இருக்கக்கூடிய லெசன் 4 பிரின்சிபிள்ஸ் அண்ட் ப்ராசஸ் ஆஃப் பயோடெக்னாலஜியில் 1 வேர்ட்ஸ் கொஸ்டின்ஸ் அண்ட் ஆன்சர்ஸ் நம்ம பார்த்துட்டு இருக்கிறோம். அந்த வரிசையில் பார்ட் 2ல் நாம் சந்திக்கிறோம். Which substance is used to cosmetic products for healthy hair and skin? SCP. Which is used as the excellent source of protein for feeding cattle, birds, fishes, etc. SCP. Which is used in food industry as aroma carriers, vitamin carriers, emulsifying agents. The same SCP. Which is used as foam stabilizers. SCP. What are the other names of genetic engineering? Recombinant DNA technology, rDNA technology, gene cloning. Which process includes different experimental protocols resulting in the modification and transfer of DNA from one organism to another? Genetic engineering. Which process involves exchange of recombination of genes between homologous chromosomes? Conventional recombination. Recombination carried out artificially using modern technology is called recombinant DNA technology, that is rDNA technology. Recombinant DNA technology is otherwise called as gene manipulation. Isolation of DNA fragment containing a gene of interest that needs to be cloned is called as insert. Abbreviate PCR. Polymerase chain reaction. Which technique is commonly used in laboratory to make copies, millions of copies of particular region of DNA? PCR. Generation of recombinant DNA molecule is done by insertion of the DNA fragment. What are the basic tools of rDNA technology? Enzymes, vectors, and host organisms. The most important enzymes required for genetic engineering are restriction enzymes, DNA ligase, and alkaline phosphatase. The two enzymes responsible for restricting the growth of bacteriophage in E. coli were isolated in the year 1963. Which enzyme add methyl groups to DNA? DNA ligase. Which enzyme cut DNA? Restriction enzyme. Which enzyme cleaves DNA into fragments at or near specific recognition sites? Restriction enzyme or restriction endonuclease. The specific recognition sites with the DNA molecule known as restriction sites. What are the two types of restriction enzymes on the basis of mode of action? Endonuclease and exonuclease. Name the enzymes which remove nucleotides one at a time from the end of a DNA molecule. Exonuclease. Write some examples for exonucleases. BAL31, exonuclease 3. Name the enzymes which break the internal phosphodiester bonds within a DNA molecule. Endonucleases. Give some examples for endonucleases. HIND2, ECOR1, PVU1, BAMH1, TAC1. What kind of enzymes are called molecular scissors? Restriction enzymes. Name the material which acts as a foundation of recombinant DNA technology. Restriction enzymes. Name the enzymes exist in many bacteria where they function as a part of their defense mechanism. Restriction enzymes. What are the three classes of restriction endonucleases? Type 1, type 2 and type 3. Which type of restriction enzyme preferred for use of rDNA technology? Type 2. How many base pairs are present in type 2 restriction enzymes? 4 to 8 base pairs. How many base pairs of DNA will be cut with Hind2? 6. How many recognition sequences have been isolated till date? 900. From how many bacteria restriction enzymes are isolated till now? 230 strains of bacteria. The first letter of the restriction enzyme indicates the genus name. Some restriction enzymes cleave both strands of DNA through the center, resulting in blunt or flesh end. These are known as symmetric cuts. Some restriction enzymes cut in a way producing protruding and recessed ends, known as sticky or cohesive end. Sticky or cohesive end are called staggered or asymmetric cuts. Which enzyme joins the sugar and phosphate molecules of double-stranded DNA with 5' phosphate and a 3' OH in ATP-dependent reaction? DNA ligase. DNA ligase is isolated from T4 bacteriophage, which is a DNA modifying enzyme and adds or removes specific phosphate groups at 5' terminus of double-stranded DNA or single-stranded DNA or RNA. Alkaline phosphatase. Which enzyme prevents self-ligation? Alkaline phosphatase. Which enzyme is purified from bacteria and cough intestine? Alkaline phosphatase. Major component of a gene cloning experiment is called vector. Dash is a small DNA molecule capable of self-replication and is used as a carrier and transporter of DNA fragment, which is inserted into it for cloning experiments. Vector. Vector is also called cloning vehicle or cloning DNA. What are the types of vector? Cloning vector and expression vector, which is used for the cloning of DNA insert inside the suitable host cell. Cloning vector. Which type of vector is used to express the DNA insert for producing specific protein inside the host? Expression vector. What is MCS? Multiple cloning site. மேலும் பல தகவல்களை தெரிந்து கொள்வதற்கு பயாலஜி தமிழ்நாடு சேனலை சப்ஸ்கிரைப் பண்ணுங்கள். நன்றி, வணக்கம்.
பலருக்கு சேவை செஞ்ச நீயும் ஒரு ஹீரோடா. ஒரு கைப்பிடி சோறானாலும் ஷேர் செஞ்சு பாருடா.